All right. It's a pleasure to have Anastasia Yendiki, who's an associate professor in radiology and director of analysis laboratory for computational neuroimaging at the Martineau Center and Harvard Medical School. Uh, I've known Anastasia for a long time, and many of you have probably used many of the tools that she has made over the years. She keeps me on track thinking about Python tools and kits out there. But over the years, she's kind of transformed her love for computation, techniques, and brain imaging into this new project that she's leading called Link, which is part of the Brain Initiative Brain Connects program, which brings together understanding anatomy at scales from cells to circuits. With that, I'm going to let Anastasia talk about things, and I think any interruptions for quick clarifications are okay.
Absolutely, yeah.
And we'll leave discussions or bigger questions for the end.
Sure, yeah.
Thanks, Anastasia.
If thank you for the lovely introduction, Sadra, and thank you to the organizers for inviting me. Yeah, as Sadra said, feel free to jump in and please someone shout at me if there's a hand up and I don't see it. I'll keep the gallery on the side. Yeah, so this talk is going to be about first about why we might want to do a crazy thing like that, scale human neuroimaging from the scales that we're used to mesoscopic and microscopic resolution. And then what are the technical challenges and the areas where work from this community will be most welcome. So to this day, most of what we know about how different parts of the brain are connected to each other come from two different sources of information, very classical anatomic kind of techniques. One is gross dissection, where an anatomist goes and takes a postmortem brain and they freeze and thaw it and freeze and thaw it iteratively, which makes the fiber bundles easier to peel apart physically. And this has a bunch of issues, of course. It's a distractive technique. You cannot get a full brain connectome. As you try to dissect one pathway, you have to destroy the ones that are crossing with it. It can only give us the sort of high level information, but it is feasible in human brains. The other sorts of information that most of our knowledge of what's connected to what in the brain comes from is tracer injection studies, which cannot be done in humans. They are done in non-human primates, macaque monkeys, typically. And these have very high precision. So we can see every individual fiber from the place where the tracer was injected in the gray matter to traveling through the white matter and to all the terminations that one injection site is connected to. But it also gives us incomplete information. So we can, with these classical techniques, do sort of a handful of different injections in different parts of the brain with different tracers that don't interact with each other. So again, we cannot get a full brain connectome, and it cannot be done in humans. In parallel with these, classical techniques that actually most of what a neurosurgeon would use when they, are about to cut open your brain and they need to know what to cut or not to cut, depending on what's connected to what, comes from those classical anatomic techniques. But in parallel, over the last few decades, we've had the introduction of diffusion MRI tractography, which is a way that allows us with just a quick scan, unlike those other classical techniques that are very time consuming and very laborious. With diffusion MRI, with a quick, let's say 10-minute scan, we can get a whole brain connectome from a living human subject. It gives us information about the full connectivity of the brain, but of course it has its own limitations. So it's known not to have the accuracy of these classical anatomic, the invasive techniques. However, it's the only technique that we have to date to be able to image the brain connections in a living patient, which is helpful for several different reasons. So, of course, it's helpful in neuroscientific studies of looking at the maturation of these pathways in brain development, or how they change with aging or different disorders. And in terms of clinical applications, getting these connections right is very important for targeting the right connections for therapeutic interventions with neuromodulation. And an example of a neuromodulation technique that relies on targeting the right white matter connections is deep brain stimulation. And there are different sweet spots that have been identified over the years for both motor disorders like Parkinson's and tremor, and at a more experimental level, also for psychiatric disorders, including major depression and obsessive compulsive disorder. So one thing that has come out of the studies that have been done so far using the sort of imperfect human in vivo neuroimaging techniques that we have with fusion MRI is that it's important to know exactly where depending on how the electrode is implanted, it's important to know which exact fibers it's going to activate. And getting that with high precision is important both for getting the therapeutic effect that we desire to have for the right disorder that the patient has, and also for avoiding side effects. So it's known, for example, that with Parkinson's disease, if you do not exactly hit the fibers that you need to hit and hit some additional fibers, it can lead to depression symptoms, for example, in patients that didn't have those kinds of symptoms before. So because of that, it's really important to get these fibers right. These fibers typically are very long-range connections that connect the subcortical structures that are being stimulated all the way to the cortex. And so they are really challenging connections to image with diffusion MRI. And so to this date, most of what we know about how these structures are connected to each other comes still from those classical techniques. From the tracer studies in non-human primates and the postmortem dissections. And so to this day, the best models that exist out there to model deep brain stimulation to better understand its effects as well as to target it actually comes not from diffusion tractography, because that's not it's considered not reliable enough to dissect these specific cortical, subcortical, and small within the subcortex pathways that are important for these disorders. But they actually come based on information purely from animal studies. So one of the state-of-the-art models is shown here from Peterson et al. From Cameron McIntyre's group. This was created based on synthetic fibers, not based on diffusion tractography. And the way this was created is that they brought together a bunch of neuroanatomists. My close collaborator, Suzanne Haber, is shown here in 3D goggles. And based on their knowledge from the tracer injection studies, they had them recreate what these fibers would look like in the human brain based on the injection studies in macaques. And this kind of synthetic atlas actually to this day seems to perform better than real diffusion MRI data in terms of predicting, being able to predict symptom improvement depending on the location where the electrode has been placed. So clearly we have a gap with our in vivo neuroimaging technologies. And the major problem here is that we do not have ground truth. So of course, there have been, lots of advances with artificial intelligence and machine learning, and those techniques need to learn from something. So unlike, when the situation where we can download, 1,000 images of cats and 1,000 images of dogs from a Google search and train a model that can separate the two, with these kinds of things, we don't have, 1,000 examples of correct fiber pathways in the brain, or even fewer than those. So that we cannot take advantage of all the latest techniques to improve our ability to image these pathways directly in individual patients because we don't have the ground truth data to train these techniques on. However, there has been over the past 10 and even more years, lots of efforts within the diffusion MRI community to better understand where does tractography go wrong and to compare it side by side with different microscopy techniques. And this is how I got sucked into this madness of going from in vivo neuroimaging all the way down to the microscopic scale. I won't go into all the different work that has been done in this domain, validating diffusion MRI against microscopy, but you can check out this review paper that we wrote for the special issue on the 10 years of the Human Connectome Project in our formerly favorite journal, Neuroimage, a few years ago. So how I started doing this kind of validation work is working with Suzanne Haber, who's a well-known neuroanatomist doing primate work with tracer injections. And she sent us brains from macaques that had received tracer injections in her lab. We take the postmortem brain, we do a weekend-long scan, collecting diffusion MRI data for 48 plus hours here in a small bore scanner here at the Martino Center. And then we send it back to Suzanne, where her lab does the sectioning and the staining to visualize the tracers that are inside these brains. And then we can do a side-by-side comparison of the tractography and the tracers in the same animal. And we've done some of our own studies, but also we made this some of these data publicly available as part of an open tractography challenge that we organized a few years ago. That's the Iron Track Challenge, where teams of diffusion tractography developers from all around the world optimized their pipeline, starting all the way from pre-processing to tractography, by comparing their accuracy of the tractography against the tracer injection pathways inside one brain, and then applied the optimized pipeline to another injection site and another brain to see how robust that optimization and the optimal parameters were when we were looking at seeding in different parts of the brain. Yes, Julie,
Yeah.
there's a question?
Sorry, I did have a quick question. It goes back just a little bit, but just to make sure I understood, you actually do the whole brain macaque scan first, right? And then it gets sectioned.
Exactly.
Okay, I just want to make sure that was.
Yeah. So they do the injection, then they perfuse the animal, and after the brain's fixed, it gets sent to us. We image it, and then we send it back, and then they section and stain. Yeah.
Thank you.
No problem. So some of the outcomes of that earlier effort was we were able to recommend best practices in terms of analyzing diffusion MRI data to achieve optimal accuracy of tractography with respect to anatomic tracing. Best practices in terms of the pre-processing, the orientation reconstruction, and the tractography methods. And also we identified some common failure modes across all the state-of-the-art at that time diffusion MRI tractography pipelines. So we asked, what are the parts of the brain where pretty much all the methods or most of the methods get it wrong? And what is special about the fiber configurations in those parts of the brains? And so that was also a very helpful exercise because we found out something quite interesting, that although the community has been focused very much on resolving the infamous crossing fiber problem, actually it seems like we are doing pretty well in terms of resolving crossing fibers. And now there's everything else that makes tractography go wrong. So fiber configurations other than crossing, such as branching, fanning, turning, etc., because all the crossing fiber models basically turn the fiber configurations in a voxel into a crossing. So this is just a few examples of how comparing the neuroimaging data to microscopy can give us insights that would be hard to gain elsewhere. So just to give you some examples of why this is important. So branching is actually pretty much everywhere in the brain. So we haven't paid too much attention to it until recently. But if you think about it, every part of the brain is connected to multiple other parts of the brain. So in order to go from one to many, you have to have some branch points. And so these are actually pretty common around the brain, and you can see them very clearly in the tracer injection data, where one large group of fibers that had just come out of the injection site then splits into multiple smaller bundles. So if we model this kind of thing as a crossing, obviously we would get things wrong. Then another interesting insight that we gained from this kind of exercise of comparing the diffusion tractography to the tracer data, and that also kind of flips some of the assumptions that we make as tractographers on their head, is that what we call a fiber bundle is not as easy to define as we might think. So we think of, lots of fibers that are starting from the same point, travel parallel to each other and end at the same point. But we find that that model is sometimes what we see under the microscope. So this is an example of fibers that all originated in the same injection site as they're traveling through the internal capsule. And we see that they stay very, very close to each other, very bundled as they travel, far from their point of origin. Then if we look at fibers from that same injection site as they travel through the corpus callosum, again, they tend to all travel through the same part of the corpus callosum. But if we look, for example, inside the uncinate fasciculus, we see that these fibers that came from the same injection site in the cortex tend to basically be spread out throughout this large highway. And so this is important for things like microstructure-informed tractography, for example, which is one approach that people have come up with in the tractography community with the goal of trying to resolve all these different kinds of fiber configurations. But this assumes that fibers that are close to each other actually have similar microstructure because they all belong to the same bundle. But in some cases, what we see under the microscope actually disagrees with that and shows us that there are fibers that are, close to each other, but are probably going to come from different cortical areas, because in all these gaps that you see here between the fibers that came from that one injection site, there are fibers that are coming from different injection sites. So just to give you an idea of why doing this kind of exercise is important for going back and developing better tools for analyzing neuroimaging data. So all that stuff was in monkeys, and then, obviously we wanted to do something in the human brain. We like humans. And so in our lab, we have been collaborating with the optics team here at the Martino Center, and they have been working on a technique that's called polarization-sensitive optical coherence tomography. I'm going to show you a little bit more about that later. But just to show you a study that was done by Robert Jones, who was an analyst in our group, a few years ago, comparing diffusion orientations to direct measurements from of axonal orientations from optical coherence tomography, again with the goal of comparing different diffusion orientation reconstruction methods and figuring out which one is the most accurate. But now this was done in small human samples. At the time, we didn't have a big microtome, so we could cut whole human hemispheres like we have now, as I'll show you in a little bit. But so then it was limited to a smaller area within the white matter. So we picked this area that's interesting because it has fibers that are splitting between the internal and the external capsule. There's crossing. There's all sorts of things going on. We collected the data so that we could generate data sets with different diffusion sampling schemes, so single shell, multi-shell, as well as Cartesian for DSI. And then we looked at what happens when we downsample the data from their original resolution of 0.25 millimeter isotropic down to something that would be more common in vivo, like 2 millimeter isotropic. And then these data were compared directly to polarization-sensitive optical coherence tomography collected from the same sample. So what you see here is a direct measurement of the in-plane orientation of fibers from this sample as it comes out of the PSO-CT scan. So it does not require any further processing as it's a direct measurement of this kind of optical imaging technique. And so interestingly, what Robert found was that when he compared different factors in terms of what made the biggest impact as far as the accuracy of the diffusion orientations when compared to the optical measurements, he found that the spatial resolution of the diffusion data played the biggest role, followed by which method was used to reconstruct the orientations from the diffusion data, followed by which sampling scheme, single or multi-shell, et cetera, was used. Interestingly, he found that the biggest jump or the biggest loss in accuracy was when we went from 1 millimeter to 2 millimeter resolution. And that's a hopeful message because there is quite a bit of work going on in vivo neuroimaging right now to reduce the voxel size that we can acquire in vivo. And so this is sort of a feasible goal to maybe one day soon be able to go down to 1 millimeter resolution. And a bit echoing the results that we had seen from the tracer injection studies in the macaque, Robert found that actually the areas that were trickiest, oops, sorry about that. I'm trying to get rid of my Zoom thing. Okay. It does not want to go away. Sorry about that. Okay. So again, he found that the reason that there was a big drop in accuracy as we went from 1 millimeter to 2 millimeter resolution was that these branch points were the ones that were hardest to deal with and caused the most errors, the parts where fibers were branching as opposed to crossing when we went to the lowest spatial resolution. And that was a problem for basically any kind of orientation reconstruction method. It didn't matter which kind of way you had to resolve crossing fibers. When you have this branch point, they were all failing. So this is just some examples of things that we've learned from these sort of smaller validation studies of diffusion MRI against these different microscopy techniques. So we started, slowly building up and trying to do this at a larger scale. But remember, at that time, we could only do the optical imaging in small blocks. So this is an effort that was funded by a previously funded R01 project of mine and led by Chiara Maffei in our group to follow the fibers in the superior longitudinal fasciculus in postmortem brains by scanning multiple blocks cut out of the same human hemispheres. And we picked this fiber bundle because it's one that has a lot of controversy around it in tractography studies, as well as in previous anatomical studies. So there are studies that say that this pathway goes all the way from the frontal to the parietal cortex, others that say that it terminates earlier before it reaches all the way to the frontal cortex. And so it's quite controversial. What we expected was that actually as we would go to higher and higher resolutions, we would see more of these longer fibers going all the way from frontal to parietal. And we thought that it's because of the lowest resolution, you have a lot of crossing fibers from the motor near the motor cortex area or from the corpus callosum. And so these get interrupted sometimes and you don't see the whole long range connection. But actually what we found when we went to the higher and higher resolutions, both first scanning these blocks with high resolution diffusion MRI and then scanning them with optical imaging, down to a resolution of a few micron, is that actually at high resolutions, we see more of these shorter range connections. And this is not surprising to an anatomist because previous tracer studies in non-human primates have described this, basically describing how these large pathways that in tractography we think of them as one monolithic structure, they are actually more like a highway that serves as a conduit for smaller fiber bundles that merge onto the highway, travel along it for part of its trajectory, and then merge off and connect a more nearby cortical area. And so we found consistently as we went to the highest resolutions that actually these, what we think of as long-range association pathways may actually be composed of many smaller, shorter range connections. This is work that Chiara is working on preparing for publication quite soon. So we think this is important because it reflects on a lot of the work that we do automating the reconstruction of these pathways, because in tractography, we typically think of these pathways as just one thing that goes from point A to point B. But actually, if you think about it, each of these pathways is its own connectivity matrix. And so that also has implications for how we understand the function that is subserved by these pathways, and also how we understand alterations in different points along this pathway that we see in disease. So then we wanted to do more of this, and that's how we get to the bigger boat. So it was a lucky coincidence that while we were working on this validation work, the Brain Initiative came out with an announcement for the Brain Connects program, which was a $150 million total investment to produce wiring diagrams across many species, from the mouse to the marmoset, macaque, and human, at unprecedented resolutions. And so this funded 5 UM1 comprehensive centers, 2 in the mouse, 1 in the marmoset, and 2 in the macaque and human, as well as a few more smaller UO1 specialized projects. And we were lucky to receive one of the 2 centers that are focused on the macaque and human brain. And this is the project that Satra mentioned earlier, and that he's a big part of building the data infrastructure for. So the project is called Large Scale Imaging of Neural Circuits, or LINC. My co-PIs on this are Suzanne Haber, my long-term neuroanatomist collaborator, and Elizabeth Hillman, who is a wizard of light sheet microscopy. You'll see some of her beautiful pictures later. And so, as I said, we're focusing on the macaque and human brain, and we're going all the way from collecting from sub-millimeter resolution diffusion MRI to sub-micron resolution data with both optical and X-ray microscopy. I'll show you some of these novel microscopy techniques that different teams on the project are going to be using. And our goal goes back to that application that I showed you earlier of getting a more accurate model of the fiber pathways that are relevant to deep brain stimulation. So the goal is to image cortico-subcortical projections that are relevant to deep brain stimulation for both motor and psychiatric disorders. So this problem, there's competing demands here. So there's one demand for large field of view, because we have to follow fibers all the way from the cortex to the subcortex. So there's no way to do this in a small chunk of brain anymore. And in addition to that, we need ultra high resolution in areas of the brain where you have many of these fibers going through being packed through very narrow passages, like the internal capsule that contains fibers from pretty much almost everywhere in the cortex. So if we try to satisfy both of those requirements, both the large field of view and the ultra high resolution at the same time, things will become intractable in terms of data storage, transfer, computation, visualization, anything. And so because of that, we approach this problem with a multimodal, multi-scale imaging approach that involves 3 different optical and X-ray microscopy techniques, as well as the mesoscopic diffusion MRI to link it back to in vivo neuroimaging and to clinical applications. In the macaque, we'll have tracer injections in these brains done in Suzanne's lab at the University of Rochester that will serve as the ground truth to validate the more novel microscopy techniques that will be applied to these brains downstream. Because in the human, we're not going to have such a hardcore ground truth, because we cannot do tracer injections, we use the macaque as a model to be able to do the direct validation of these methods against the tracer injections, and then to apply these methods to the human brain. In the human brain, we also know that there are certain topographic organizations of these fibers that we can also use as sort of an indirect way to replicate, trying to replicate these topographic organizations in the human brain as a way to sort of verify that our methods are working. But in the macaque, we'll have the direct validation against the tracers. In terms of the diffusion MRI, we're scanning both the macaque brains and the human brains in scanners with ultra-high gradient strength that allow us to go to very high B values as well as high spatial resolutions. This is in collaboration with Susie Huang, who was the PI of the Connectome 2.0 project that was responsible for designing and installing this new scanner, the Connectome 2.0 3 Tesla scanner here at MGH. And so we're doing the most comprehensive sampling of the whole parameter space of diffusion MRI that includes echo times, B values, diffusion times, et cetera, in using protocols that have been optimized both for orientation reconstruction as well as microstructure modeling. And this is basically expanding on our work from doing the whole brain diffusion MRI scans in Suzanne's macaque brains, but now to the human brain as well. And this is just to give you a preview of what these human hemispheres that have been scanned on the Connectome 2.0 look like. So this is a 400 micron isotropic resolution scan of a human hemisphere. This was a protocol that was developed by Ting Gong that you see down here, who's a postdoc in our lab, and Dong Suk Sang also helped with the acquisition. He's a postdoc in Susie's lab. So these are data that we are preparing for distribution and hopefully will be distributed through Dandy later this year. And we're preparing a preprint to go with that. So this is the highest resolution whole hemisphere diffusion scan in the human brain to date. And in addition to this protocol, where the goal was to go to as high resolution as possible for tractography to identify small subcortical bundles, we also have a couple of protocols that are optimized for microstructural modeling. For example, a multi-diffusion time diffusion MRI protocol to allow us to estimate axon diameter indices. And you see the nice differentiation here of axonal diameters in this whole hemisphere scan. This is at a somewhat lower spatial resolution of 800 microns in order to make it feasible to collect the multiple diffusion times and the higher B values that are needed for this kind of analysis that was done by Ting using her newly developed package of microstructure.jl. And in addition to this protocol, we have a multi-echo time protocol for doing diffusion relaxometry and being able to look at diffusion measures in the gray matter as well as the white matter. So this kind of modeling allows us to look at things like cell soma fraction and neurite fraction in the cortex, and be able to estimate cell soma and neurite specific T2, as well as soma radius. So this was another protocol that was involved multiple echo times as well as multiple B values at a somewhat lower spatial resolution of 1 millimeter. So all these three protocols, we have them in two hemispheres, and we have other hemispheres in the pipeline to be scanned. So what will happen to these hemispheres, both the macaque and the human ones, afterwards, after a diffusion MRI? The first step is to collect the data that I showed you earlier, the polarization sensitive optical coherence tomography data, or PSOCT, that involve direct measurements of 3D axonal orientations at microscopic resolutions. But now not in small samples, but in whole hemispheres. And so this is work that is done by, led by Hui Wang here at the Martino Center. The benefit of the PSOCT approach is that it images the block face of the tissue before cutting it. And because of that, it avoids any sort of nonlinear distortions that you get because of sectioning. So as you can see in this video here, the images from the different sections, because you image first and then you cut, come out undistorted and do not require registration to be aligned to each other. However, this does not get down to the single axon resolution still. And so that's where Elizabeth Hillman comes in. And particularly for the macaque, the block face imaging is going to be integrated with simultaneous light sheet microscopy to image the fluorescent tracers that are going to be our ground truth in the macaque. And then the sections from both the human and from the macaque are going to go to Elizabeth's lab for further imaging. This is just to show you that we're now able to collect this kind of data in whole human hemispheres. And in addition to that, by collecting images with multiple light incidence angles, we can collect multiple measurements of the in-plane axonal orientations that then allow us to infer the three-dimensional axonal orientation, thanks to work by Chao Lu, who was a postdoc in Hui's lab a few years ago. So after this, the sectioning, then Zhuhao, who is going to be responsible for clearing of the tissue sections and immunolabeling with a variety of axonal markers. And then these sections will undergo light sheet microscopy at Elizabeth's lab. Now she's not at Columbia, she's at St. Jude. I should fix that. To go down to sub-micron resolution, where we will be able to image individual axons. And this is just an example showing that this is based on work that Zhuhao and Elizabeth were doing before this project as part of their BICCN cell census network project that involved developing tools for human brain clearing, as well as ultra-fast spectrally multiplexed 3D imaging to allow imaging with light sheet microscopy of multiple stains at the same time. And this is just an example of data from one of the axonal markers that we're testing for our project. These were collected by Amina, who's a grad student in Elizabeth's lab, showing our ability to image individual fibers. So this is what you see on the left is the actual microscopy image. It's a maximum intensity projections through a section. And what you see on the right is fiber orientations that were extracted from a simple structure tensor analysis, where basically we're just finding the gradient of the microscopy image in different directions. And this allows us to then just run tractography on these orientation vectors, similarly to how we would do it with diffusion MRI data, and to reconstruct the trajectories of individual fibers. In addition to these optical imaging techniques, last but not least, we have an innovative X-ray microscopy technique called HIP-CT, or hierarchical phase contrast tomography, that has been pioneered by Claire Walsh and Peter Lee at UCL. And it allows label-free, without any staining, imaging of entire organs, so even entire human brains, in a high-energy synchrotron source at resolutions that can range from 200 micron in the whole brain down to a few micron in small areas within the intact brain. And this will be validated against conventional osmium-stained, micro-CT of osmium-stained sections in small areas within the brain to validate the more novel HIP-CT contrast. So again, I don't know if you've been counting, this is like about 5 different modalities that we'll have to put together. This is an example from previous work by Claire and Peter. These are data that are collected only in one place in the world, the European Synchrotron Radiation Facility in the French Alps, showing how in the same brain, they can collect an overview scan, and then they can zoom into cylinders within the brain and collect data with just a resolution of a couple of microns. And because they are collected within the intact brain, they are all these data sets at the different scales are inherently aligned with each other. They're collected in the same session, allowing for truly multi-scale imaging all the way down to the cellular level. So you can see here the little cylinders with different colors where they did higher and higher resolution scans. And so now we have one of those brains that got that beautiful diffusion scan, has also gotten the full HIP-CT protocol, and we're in the process of putting all that data together for publication later this year, hopefully, and for public release. And this just to show you that if you cut small samples from the brain, then you can go down to submicron resolution with HIP-CT. Of course, this cannot be scaled up to the whole brain, but in individual areas of interest, we can go down to resolutions where we may be able to identify even individual fibers. Okay, so then all of these data are going to come back to us. We're the computational team, Satra, Kabi from Satra's team, and Yael, who used to be at MGH and now is at UCL, have been the key MVP players in this effort. So we have our work cut out for us because we have to develop methods for registering all these different data from the different modalities and the different scales to each other within a brain and across different brains, develop tools to segment the axons from the different modalities and to reconstruct axonal trajectories, to reconstruct accurate wiring diagrams of the brain. And then, of course, there's the big question of how do you disseminate these huge data sets? How do you make them easily accessible for remote visualization and remote computation, as well as building easy tools to be able to use these kinds of data to validate in vivo neuroimaging techniques. So that's some of the work that we have been doing together with Satra in the last couple of years. And so just to give you a brief overview of some of the methods that we're developing for the analysis, one is developing deep learning models for segmentation of anatomic tracing data, so finding these fibers and reconstructing fiber bundles from the histological sections. This was work that was started by Vanathi Sundarasan, who was a postdoc in our group a couple of years ago, published in our new favorite journal, Imaging Neuroscience. And now this work is being taken further by Margarita, who's a postdoc who started in our lab this year. And so we continue to develop this work to use more modern architectures than what was available 2 or 3 years ago, as well as use synthesis techniques to augment the training data, and so on and so forth. So this is ongoing work that Margarita is actually presenting at CDMRI later this month. And so what are we going to need? So we're going to need a lot of things. First of all, we're going to need appropriate data formats. We cannot use Nifty anymore, unfortunately. So the goal here is to go from images that have millions of voxels, so that's the typical 256 cubed neuroimaging volume, to billions of voxels. So this is one histology section at submicron resolution, contains 13 billion pixels, just to give you an idea. So the neuroimaging formats that we know and love, like Nifty, they have a lot of great features. So Nifty, for example, includes the transform from the voxel to the world coordinate system, which is so important, particularly when you're combining different data sets and for visualization. However, typically it's used in use cases where the image can be loaded in full in memory. The microscopy technique, the microscopy community, on the other hand, has been developing techniques to deal with these huge data sets where you cannot load the whole image all at once. And OMESR is the current standard in terms of formats that allow the image to be loaded both at different scales of resolution and in smaller chunks. It facilitates data streaming. So when the user is viewing the whole brain like this, you can load a lower resolution version of the image, and as they zoom into a specific part of the brain, you can load the higher and higher resolution version, but only from the smaller areas that the user is viewing. One issue with this format is that it does not contain the full transform from the voxel to the world coordinate system. And so Yael has been working on a hybrid format, while in the meantime we're waiting, and hopefully the OMESR community will start including this kind of information within the OMESR data sets. And this solves our problem with loading volumetric data, but then we have other kinds of data like surfaces or streamlines in our case from tractography. And we don't have comparable chunked formats for only loading parts of streamlines when we're looking at ultra high resolution. So these are problems that we will have to solve in the next few years to be able to do visualization of all these different kinds of data sets at the same time. So just to give you an idea of work that has been spearheaded by Yael in collaboration with Satra's team. So this is using an existing visualization platform that's called NeuroGlancer, that's very popular in the nanoscale electron microscopy connectomics community. And has been developing add-ons to be able to align load the coordinate transforms from different types of data sets, be able to combine these data sets in the same scene, and be able to also load streamlines, which is not something that was a use case for this viewer for NeuroGlancer previously. So this is all work that's very much in progress, and that we have a lot to do to further develop in the next few years. And of course, then the issue of annotation, in our favorite neuroimaging viewers like FSLIs or Freeview and so on and so forth. We have taken for granted how easy it is to do these tasks. But for these kinds of microscopy data sets, these tasks are still not, they're not straightforward to do with these visualization tools. So we want to ideally have a platform that integrates the pluses of the neuroimaging visualization tools that can integrate volume, surface, and streamline data, but with the plus of the visualization tools like NeuroGlancer from the nanoscale microscopy community that can work with chunk data and are well suited to these billions rather than millions of voxels. And so clearly we need to expand some of these tools to be able to support annotation of all of these kinds of data sets, both volumetric and streamline, and to be able to read all these new formats that hopefully will be developed to store these data in the coming years. And then the analysis tools. So we know and love a lot of tools for brain registration and segmentation, but these will have to be refactored to work with these chunked data formats and to be able to operate at the appropriate scale of resolution. So for like whole brain registration, maybe we do not need the full submicron resolution. We need some intermediate scale, whereas for segmenting axons, for example, we do need the full scale of resolution. So lots of work to do and lots of food for thought, I hope. And I'm happy to hear ideas and questions about these things. Just to bring us back to why we're doing this in the first place. So the end goal is to develop the next generation of fiber models for neuromodulation and to develop the interfaces to be able to share these in a way that they're easy to use by clinicians, as well as by the neuroscientists that are interested in simulating and better understanding the effects of deep brain stimulation. So this is an example, just to give you a taste, of first iteration of this cortico-subcortical tract atlas that Chiara has been developing using those beautiful 400 micron ISO diffusion MRI data sets that we're collecting on the Connectome 2 scanner. And the colors here are based on the cortical origin of the fibers. And what we were happy to see is that we're able to replicate the topographic organizations of these fibers as they travel through the internal capsule and reach their different subcortical destination. So this is topographic organization that has been shown with tracer injections before in macaques, but has been so far very difficult to replicate with good definition in the human. So the colors at the top, as I said, are based on the cortical origin, and in the bottom row on the right, you see that Chiara has been able to nicely separate the fibers that terminate in the thalamus versus the brainstem, which is important for a lot of these neuromodulation techniques. So this is, again, something that we hope to have released by later this year. And I will end with just a shameless plug of our new favorite journal. Also something that was announced just like yesterday, I think, is that our article processing charge has gone down from 1,600 to 1,400 because the journal has been doing so well thanks to the support of the community. And if you're a diffusion person, a special announcement, we're going to have a special issue opening for submission later this fall on the past, present, and future of diffusion MRI. It was inspired by the workshop on the 40 years of diffusion MRI that was organized earlier this year. And if you want more information, you can contact Santiago and Nemi, that are the editors of the special issue. And I will end by thanking the Link team. Lots of good times that you see here. This is all from when the grant first got funded, and so everybody was happy and relaxed. And now it's everybody's like exhausted and stressed out, and we need a new set of pictures to update this. But I will end here, and I'm happy to take questions. I don't know how much. Oh, didn't leave a lot of time. Sorry about that.
No worries. Thank you very much.
Is anyone left awake? Oh, JB's clapping. Thank you.
Yeah, a silly question.
Yes,
Probably,
please.
how much disk space exactly does one of those huge scan files take up?
Yeah, that's what we're trying to figure out. So one of those OCT scans from like one slab, like about 2 centimeter slab of human brain, takes like about 80 terabytes. just to give you an idea. And that's not the highest resolution. That's like the one that's about like 6-micron resolution. So the light sheet microscopy ones that go down to sub-micron scale are obviously even bigger. And so a big question that comes out of that is like, do we need to go down to the highest scale everywhere in the brain? Or can we somehow like smartly sample more sparsely at the highest scales of resolution and combine with the low resolution data? And so, this is something for, that would be a great thesis or postdoc project for someone to work on.
Cool, thanks.
Maybe one quick silly question. How much of the previous research on that topic do you think you're going to invalidate? With,
I'm sorry, how much of the previous research on what?
On the topic of identifying those tracks, are you going to invalidate? How much of that previous research was good enough in terms of resolution things? Or
I would say we're going to augment it rather than invalidate it.
That's the political sort of answer, but
so there are some controversies that I hope that we're going to resolve. So for some of these deep brain stimulation techniques, there are multiple different sweet spots. There are different groups. There are, each of them has, the best one. And according to Suzanne, a lot of these are actually probably coming down to the same fibers. But this has been difficult to test in the human brain so far. So I think that these kinds of controversies, we hope to be able to provide the data to be able to resolve. And then just, increase the level of detail that's available. Because I showed that the Peterson Atlas with the synthetic fibers, that it has a lot of gaps in between. There's a lot of the smaller bundles that are not there. So we're hoping to fill out that space with new knowledge.
awesome.
Thanks.
Julie?
That's a question.
Yeah, well, I did have a question that was actually somewhat related to that, and I almost asked it earlier, and then I thought, you just have so many beautiful things going on here. But I kind of come back to something that does relate to, I think, what JP is asking about, which is, how is there an issue or a concern or a adaptation that may be required for dealing with subcortical structures, particularly coming down towards the brainstem, because it's a big difference in my old school experience of this between MRI and cortical, any kind of single unit tracing in the non-human primate, because you've got artifact from bone and things in at least some of the MRI scans. So it's partly a methods question, and it's partly a methods questions about imaging, and it's partly a methods question about comparing imaging with anatomy in a format that doesn't have that type of constraint.
Yeah, so there's a lot that is challenging for these areas. So first of all, they're deep in the brain, so the SNR is always lower. So even with our highest resolution in vivo data, that the cortical connections tend to be easier to study than some of these, especially the brainstem connections, etc. And then when it's in vivo, there's also other physiological artifacts, etc. So yeah, I'm not saying that we're going to invalidate everything, because some of the stuff that has been shown in vivo actually does agree with the tracer literature.
Yeah.
And if anything, I would say that when after started working with Suzanne and comparing tractography to the tracer data, I was actually surprised by how much we get right. So I don't think that it's all artifacts and it's all going to come out.
No, I'm sure it's not all artifacts. It's kind of just curiosity of, is that going to be, a component of it that has to come into it? I don't know the answer to that. I was just asking because they're obviously beautiful images now and data that you can get at resolutions that, nobody could even imagine not that long ago. So,
right. Yeah, open question. Satra, do we have a big enough boat? What do you think?
The boat is never big enough. I think some of the challenges are we're still dealing with a lack of ground truth, I think, in that in vivo space, and the number of samples we can process in the ex vivo space is going to be limited, and kind of the variation part of this is still going to be a question mark that I think will require future work to channel. But I know we're past time, so I'm going to ask, are there any other questions? Please speak up.
Two people survived in the smaller boat.
I watched the movie recently. Only one died to survive, so.
Well, thank you very much, Anastasia.
On that
note, this has been fantastic.
Yeah, thank you.
I know there's a
Thanks,
lot of work
everyone.
cut out for us
I
as
know.
people who represent this kind of information through various lenses and try to make it accessible. So thank you for pushing
Lots of
us along.
space for more people to make contributions. So yeah,
Yes.
jump on the boat.